Hello students, welcome to EPG Pathshala. I am Dr. Bibhu Prasad Panda from Jamia Hamdard, New Delhi. Today we are going to discuss about a module on essay of biological introduction and overview under paper pharmaceutical and biological analysis. In this module we will learn about what is a biological product, source for biological products, type of biological products, how our biological products are different assay systems for biologicals. Biological products definition. The term biological products means a virus, therapeutic serum, toxin, antitoxin, vaccine, blood component or derivative, or allergenic products, proteins, except any chemically synthesized polypeptides or analogs products applicable to the prevention, treatment or cure of a disease or condition of human beings. Example of biological products. Biological products are used for a wide range of diseases and conditions, including serious and life-threatening conditions such as cancer and rheumatoid arthritis. Some examples of biological products and examples of their approved use include Botox has both dermatologic and neurologic use. Herpestin for certain type of breast cancers, vaccines for example for different prevention of different bacterial or viral diseases, Enbrel for rheumatoid arthritis and psoriasis. What is a biological product? Biological products or biologics are medical products. Biological products could be made of sugar, proteins or nucleic acid or complex combination of these substances, or may be living entities such as cells and tissues. Like drug, biological products are used to either treat or cure disease and medical conditions. It may prevent diseases. It can be also used for diagnosis of disease. Biological products are made up of from a variety of natural sources. Source of biological products. Now where from we are getting the biological products that are mainly from mammalian cell culture. It may be from humans, it may be from mice or rat, it may be from microorganisms like bacteria. Biological products are also obtained from plant cell culture, from yeast, from insect cell culture, can be also obtained from transgenic animals and transgenic plants, avian cell culture, or it may be from organ culture. Type of biological products. The biological products are of various types that includes blood derivatives, whole blood components, proteins, human tissue, xenotransplantation product, cellular and gene therapy products, allergic extract, vaccines, that is maybe for preventive or for therapeutic purpose. How biological products are differ from other pharmaceutical products. We can differentiate small molecule drugs and biologicals in this way. Small molecule drugs or synthetic drugs are generally low molecular weight. However, the biologicals are generally having high molecular weights. Small molecule drugs are obtained by organic or by chemical synthesis route, whereas the biological products are obtained from live cells or from microorganisms. Small molecule drugs are synthesized with very few critical process steps, whereas the biologicals are obtained with many critical process steps through complex process. Small molecule drugs are well characterized, that means it is well characterized chemically, whereas biologicals are less characterized, that means the chemical nature of the biologicals are not easily or less characterized. Small molecules are having known structure, that is molecular structure, whereas the biologicals, the structures may be or may not be completely defined or known. The small molecule drugs are homogeneous drug substances, whereas the biologicals are heterogeneous mixture. Small molecule drugs are not immunogenic, whereas the biological, since they are mostly proteins, they often cause immunogenics or immunogenic reactions. Size and complexity of proteins. Now you can consider two molecules, that is aspirin and monoclonal antibodies. You can see that aspirin having only 180 Dalton. It is a small molecule synthesized by chemical process and very simple chemical structure, whereas the monoclonal antibodies are called MAB or MAB, having high molecular weight, that is near about 1,50,000 Dalton. It is a biological product. The monoclonal antibodies is a protein in nature and it is produced by cell culture it is produced by animal cell culture using in vitro techniques. The monoclonal antibodies is very complex. The structure of the monoclonal antibody is very complex and very large structure. Examples of a biotechnological process. The biologicals are produced mostly by biotechnological process, in which the fermenter is the vessel or the bioreactor is the vessel where we culture the live cells which produce the biologicals. The biologicals may be extracellular or it may be intracellular. After the fermentation is over, the products were harvested by downstream process using different chromatography column. The concentrated products now formulated and filled and used for human beings. Used by human beings. Biological assay. An assay is an integrative analytic procedure in laboratory medicine, pharmacology, environmental biology, and molecular biology for qualitative assessing or quantitatively measuring the presence or amount or the functional activity of a target entity or the analytes, which can be a drug or biochemical substance, organic samples, or it may be biologicals. Different type of assay we follow, that is normal chemical assay, it may be immunoassay or it may be bioassay. The type of assay depend upon the what type of molecule we are going to analyze. Chemical assay, it is the study of the separation, identification and quantification of the chemical components of natural and artificial materials. Immunoassay is a technique that can be used of the binding between an antigen and its homologous antibody in order to identify and quantify the specific antigen or antibody in a sample. Whereas in biological assay, the biological testing procedure for estimating the concentration of a pharmaceutical drug substances in a formulated drug products or in bulk material, the specific potency of the drug is given to animal or human volunteer, and then the drug response is compared with the standard drugs. Bioassay is less precise. Bioassay is also more time consuming, more expensive. Bioassay is used when active constituents and structure are not known. Bioassay is more sensitive. It requires more manpower, and also it is very difficult to handle bioassay procedure. Biological assay. Biological assay may be in vitro, or it may be in vivo, or it may be ex vivo. Under in vitro, immunological assay are most commonly used biological assays. The immunological assays includes ELISA, which is of two types. One is a double antibody sandwich, that is called DAS ELISA, or it may be DAC ELISA, that is a direct antigen coating. ELISA test is used for quantification purpose for antigen, antibody, or any protein molecules. Immunological assay also includes radioimmunoassay, that is called RIA, for antigens and biomolecules, radial immunoassay, immunoprecipitation assay, agglutination test, Coombs test, radial immunodiffusion, or it include also immunoelectrophoresis. In vitro assay or in vitro biological assay is carried out in isolated cells, that is by using different cell lines of cancerous origin or non-cancerous cell lines. In vitro biological assay also can be carried out in isolated or purified enzymes. In vitro biological assay also can be carried out in cultured organs or in cultured tissue system, whereas the biological assay, which is ex vivo assay, is carried out in isolated organs or tissue from animal. Indications of bioassay. Number 1, chemical method is either not available, if available too complex or insensitive to low dose, example histamine. Number 2, if active principle of drug is not known, that is for example insulin. Third, number 3, the unknown chemical composition, for example long acting thyroid stimulator. Chemical composition of drug variable but has some pharmacological action, for example cardiac glycosides. Active principles cannot be isolated, for example posterior pituitary extract, that is insulin, or similar like compounds. Biological activity of drug cannot defined by a chemical assay, for example cis or and trans form of the different molecules. Not possible to separate interfering substances, for example vitamin D. Principle of bioassay. All bioassay should be comparative against a standard drug. Standard and new drug should be as far as possible identical to each other. Activity assayed should be the activity of the interest. The degree of pharmacological response produced should be reproducible under identical condition, for example adrenaline. Method of composition, preferably not essentially the test therapeutic property of drug. Individual variations must be minimized. Type of bioassays. There are mainly three types of bioassays other than qualitative assays. They include direct assays, number 2 indirect assay based on quantitative response, and third type of bioassay is indirect assay based on quantal response, that is all or none. Direct assay, the dose of the standard and test preparation are sufficient to produce a specified response and can be directly measured from the response result. Indirect assay. In indirect bioassay, the relationship between the dose and response of each preparation is first ascertained, then the dose corresponding to a given response is obtained from the relation of each preparation separately. Other classifications of bioassay include quantal assay, graded assay, effect produced in confined period. Quantal assay. In quantal assay, all or non-response in all individuals is analyzed. For example, digitalis induced cardiac arrest in guinea pigs, hypoglycemic convulsion in mice by insulin, and calculation of LD50 in mice or rats. Quantal bioassay is not precise. It is mainly employed for comparison of LD50 and ED50 and comparison of threshold response. Graded bioassay. In graded bioassay, effect or biological effect is produced gradually depending upon the dose. That means the effect of drug depend upon the dose. For example, contraction of smooth muscles preparations. Purpose of bioassay. Why there is a need to carry out bioassay? Number one, comparison of test sample with standard substance to determine quantity of test samples required to produce an equivalent biological response to that of the standard substances. Number two, measuring pharmacological activity of new or chemically undefined substances. Number three, test method employed in measuring the response of living animals to toxicity of chemical contaminant. Certain number of individuals of sensitive species are exposed to specific concentration of contaminant for specific period to examine the toxic effect. Number 4, investigating function of endogenous mediators. Number 5, determine concentration as well as potency of unknown substances, improving and maintaining and standard of basic environmental conditions affecting well-being of peoples, for example pollutants released by particular source. Number 7, to determine specificity of compound to be used. For example, penicillins are effective against gram-positive bacteria but not to gram-negative bacteria. Preparation of standard or standard substances. Preparation of how we can prepare the standard substances or standard drug. A selective representative samples of standards for which it is to serve as a basis of measurement is called standard preparation. The standard preparation must be of uniform quality and it must be stable. Type of standard preparations. There are two types of standard preparation. Number 1, international standard and reference standard, that is USP unit. And second type of standard preparation is British standard. Country wants to have its own standard preparation and then used according to its own law for biological assay and test. Units are referred called Great Britons. For specific biological activity, small quantity of standard preparations are used as reference standard. In biological assay, the potency measurement of drug activity expressed in terms of amount required to produce an effect of given intensity. For example, highly potent drugs like morphine, alprazolam, chlorpromazine, highly potent drug like morphine, alprazolam, and chlorpromazine, etc., produce high response at low concentration, and a low potent drug like ibuprofen and acetyl salicylic acid produce low response at low concentration. The activity measured in terms of affinity and efficacy of the drug towards a biological system. Advantages of biological assay include bioassay are procedures that can determine the concentration of purity or biological activity of a substance such as vitamin, hormone, plant growth factor, etc. While measuring the effect of an organism, tissue cells, enzymes, or the receptor is preparing to be compared to a standard preparation. Bioassays may be qualitative or quantitative. Qualitative bioassays are used for assessing the physical effects of a substance that may not be quantified, such as abnormal development or deformity. Quantitative bioassay involve estimation of the concentration or potency of a substance by measurement of the biological response that is produces. Quantitative bioassays are typically analyzed using the methods by biostatistics. Biological assays do not only help to determine the concentration but also the potency of the samples or drug samples. It is especially used to standardize drug, vaccine, toxins or poisons, disinfectants, antiseptics, etc., as these are all used over biological system in some or in other forms. These also help to determine the specificity of a compound to be used. For example, penicillins are effective against gram-positive but not on gram-negative. Testing of infected patient sputum help determine which antibiotic to be given for quick recovery. Certain complex compounds like vitamin B12, which cannot be analyzed by simple chemical assay techniques, can be effectively estimated by bioassay or by using biological assay system. Sometimes the chemical compositions of samples are different but have same biological activity. In that case also, the biological assay is essential. For samples where no other methods of assay are available, at that time also we can use biological assay procedures for quantitative or for qualitative analysis of the samples. Biological products like toxins, antitoxins, sera, or any immunological products or immunoproducts can be conveniently assayed, measured in minute quantity, that is in nanomolar or in picomolar quantity. Quantities of active substance can be detect active substance without prior extraction or other treatments. Disadvantages of biological assay. Apart from advantages, the biological assay systems also are having some disadvantages. The disadvantages of biological assays are key problem in variability in response. Biological assays gives different response. Sometimes it may gives intraday or interday variations also. For biological assays, large number of animals to be used. Therefore, there is a issue, that is ethicals, or there is a ethical issue. To carry out biological assay, expertise in experimental design, execution of assay, and analysis of result or data is required. Biological assays are expensive and time consuming. Biological assay results change or the activity of the biological systems or response change with time. Sometimes in biological assay, the biologicals are insensitive to test organ or to test cells or to tissues. So students, let us summarize what we have learned from this module. Biological products are diverse, complex products for the treatment, diagnosis and prevention of a broad range of common and rare diseases. The complex process for manufacturing biological product testing and for evaluation of safety and efficacy of the biological products. In contrast to most drugs that are chemically synthesized and have a known structure, most biological products are complex mixtures that are not easily identified or characterized. Healthcare professional and consumer can assure that FDA, that is Food and Drug Administration, will require licensed biological products to meet the agency's exacting standards of safety and efficacy. Comparing with chemical assay and physical assay, bioassay is less elaborate, less accurate, more troublesome, more laborious and more expensive, but it is the only assay to be done if an active principle of a drug is unknown and if it is hard to isolate. Chemical method is very complex method which required high chemical doses and chemical composition shows the pharmacological actions against the drug. Other purpose of bioassay is to standardize the preparation so that each each contains the uniform specified pharmacological activity. Bioassay serve as a pointer in the commercial production of drug when chemical assay are not available or do not suffice. From the clinical point of view, bioassay may help in the diagnosis of different conditions. The bioassay is a successful total in estimation and discovery of biological active substances and important applications in sensitivity and specificity of pharmacological applications. Thank you.